Hi, I have developed this project, VSpipe-Agent, as a Capstone project through vibe coding and the concept used in 5 days program. This tool, this agent, basically it evaluates the drugs against disease HIV. The protein that is involved in HIV is called 1HSG, which is the protein data bank ID of this protein. Protein data bank is the repository of experimentally determined structures of the proteins, and the protein are the functional part. If something goes with the goes wrong with this protein, it will results in unfavorable results, and it can result in the outbursts of any disease like HIV. So this concept I have used through already published paper, VSpipe-GUI, that I had developed using Python, but it was not agent based, so I have converted it as an agent, and the concept used in this 5 days program. So this is the repository that I created while developing this project. And you can see the architecture of this, in which it takes the input, it evaluates the input. If there are the invalid input, it explains what the valid input should look like, and it will give the user and help the user to provide the valid input. And then it calculates the Lipinski parameters, which are basically the parameters that are used to categorize the drug to be accepted by the body or not. I will explain it in simple terms. And it also gave the docking score. Docking score determine the affinity of any drug with the protein drug target. So the good docking score will have the good affinity of a drug with the drug target, and Lipinski, there are the parameters that should be within the range to be accepted by the body. And finally, after the results, the LLM will be used to interpret the results and help the user what the results should look like and how to interpret them. So these are all the details are available, how it is used, and the testing was done, and all tests were posted. So this is the reference of the concept and the paper that I've used, and I must run the demo of this to show how it look like, what the results are. The input compounds, they were indinavir, aspirin, and ibuprofen. They were docked against this protein, 1HSG. And the results will be shown in the form of docking score, and whether it complies the Lipinski criteria or not. So meanwhile, it will take some time, and meanwhile I should go to the individual files used in the project, such as the schema.py, in which it defines that the input compound must be in SMILE notation. SMILE notation is the format of a chemical compound that must be provided to be accepted by the tool and by the agent. In this workflow.py, this agent is VSpipe screening workflow. This is the description what it does, and in MCP server it actually using the tool by the agent for calculating the Lipinski parameters. So let's go to back to the terminal, and these are the results. So you can see that this is the docking score of indinavir, and this is the docking score of ibuprofen and aspirin. It is showing each values in kilocalorie per mole, and it is also giving the whether it complies the Lipinski parameters or not. And you can see that indinavir is violating one rule, because its molecular weight is greater than 500 Dalton. It should be less than 500, so it is violating this rule. And other chemical compound, they are fully compliant with the parameters, and these are the type recommendations that are useful for the user to interpret. Ibuprofen is one of the most promising candidate, because it has a good binding affinity, and it also completely Lipinski compliant parameters with 0 violations. So these are the recommendations, and so this is how it works. It gives the output in the form of a docking score, Lipinski violations, and it finally gives the recommendations. All the files used in this project, they are available in this GitHub link. And thank you for watching.